Thank you, Manon, for the kind introduction. Yes, indeed, I guess this is really a product development story first and foremost. And as any entrepreneurial company, we did start out with one mandate as VGX Pharmaceuticals. We've grown through several transactions, primarily through mergers and acquisitions. And Invio today is a fully vertically integrated vaccine product development company. This has been one of the fantastic conferences where we've been able to deal with not only product development issues, but also the other aspects of vaccine development, the manufacturing, licensure, economics, distribution, that conventional R&D folks don't get to hear and talk about. So we've addressed many of these questions through the course of the meeting. The bottom line is that there's not enough attention being paid to vaccines, and not enough in terms of funding. Everybody recognizes the need, but I guess we all need to get together to push the ball up in terms of increased vaccine acceptance, new vaccine licensure, and that really involves developers, regulatory agencies, payers, healthcare providers, and a whole community as a whole. Clearly, vaccine innovation is critical. What's emerging, it's too expensive to develop not only new drugs, also new vaccines during the conventional methodologies. Low-hanging fruit have been plucked. New technologies are needed, and in particular, to deal with the issue of divergence, and that'll be the theme of my talk. So I'll tell you about our approaches for developing universal vaccines. I'll start with flu, but also tell you how we're applying these concepts to other infectious disease targets. And then new development paradigms, a lot has been talked about introducing new vaccines. So at Invio, we've established a DNA vaccine platform that consists of the drug substance, in this case a vaccine product, which would be a DNA plasmid-based vaccine. We combine that with proprietary manufacturing and formulations. This is accomplished through our affiliate VGXI Inc. They have established a CGMP facility to produce clinical product, and then we deliver these vaccines via a proprietary delivery system based on electroporation. Just very briefly, in terms of what's known about DNA vaccines and what has been established over the last 10, 15 years of development, that there are indeed no vector-induced responses elicited by the backbone, by the DNA vector. You can repeatedly boost the vaccine so you don't get amelioration of responses as you see with some of the other vectors. You can do multiple vaccines. You can combine DNA easily into formulations. Recently, we've shown, and others have shown, that in many cases, DNA vaccines can give you equivalent or better potency than viral vectors. And critically, we've also been able to show both CD4 and CD8 T-cell responses, as well as the induction of antibody responses. There's a historically strong safety record, several manufacturing advantages, and Invio has a broad and deep patent position on the EP technology. Very briefly, electroporation, as the term implies, is the application of brief, low-energy electric pulses that create transient pores to form in the cell membrane to allow macromolecules like DNA to enter the cell membrane. Once the electric pulse is removed, the cell membrane fuses. You've got DNA plasmids inside, which then express the antigen of interest, and then the immune system takes over. So it's like a viral vector, it's like a live virus immunogen without the safety concerns with what's been observed. In terms of increased potency, here's an example where we've administered GFP-expressing plasmid into rabbit muscles. These are 1 millimeter thick vertical sections. If you don't apply electroporation, if you don't deliver the plasmid with electroporation, you don't see any expression of GFP. And if you deliver GFP plasmid with EP, you see over 1,000-fold enhancement. It's so strong that you don't even need the light box. Just on plain white light, you can see GFP expression. At Invio, we've also, we look at product development holistically. So we've started out with intramuscular delivery of DNA vaccines. These are deeper penetrating needles. You need to target a muscle which typically is 19 to 25 millimeters deep. This allows us to focus immune responses towards cellular immunity, but more importantly, you can also deliver larger volumes of vaccination. So really, depending on your end needs, you might consider EP, intramuscular EP. We've since also developed delivery systems that target the ID or sub-Q layers or purely ID, and these are what we call minimally invasive, which go as little as 3 millimeters in depth, or you can look at surface electroporation, which don't scratch, which just barely scratch the surface of the skin. And so these devices are shown here. The ID and minimally invasive and non-invasive devices are shown here. These, so in this electroporation procedure, you would inject the plasmid DNA vaccine via a standard Manteau injection, and then follow that with the application of these very shallow electric pulses. Here's a GFP expression on the back of a guinea pig skin, showing you the islands of GFP expression around where the electrodes touch. We've used these systems, the ID delivery systems, more towards prophylactic vaccine indications, and we've got a number of ongoing programs internally as well as with partners. So with that brief background, let me talk to you about flu. And I think if we took a poll in this room for what does the concept of universal mean to you, I'm sure there's going to be as many responses as there are people in this room. For us, the working definition of a universal vaccine for flu would be one that elicits a broad cross-protective immune response towards drifted and shifted strains, and I guess critically, therefore, does not require annual reformulation. And so our idea here would be to try to target seasonal epidemic coverage as well as pandemic coverage. And I guess the good news around everybody who's working with universal vaccine technology or platforms is that many countries fund flu vaccination, at least amongst the high-risk groups. The recommendations are very strong, WHO guidelines, ACIP recommendations. So from a market standpoint, it's relatively easy to make the case to develop a universal vaccine. The need, and also consequently, the need is strong because current vaccines are largely strain-specific, so new formulations are required based on the circulating strains. They're not necessarily as effective against newly emergent or shifted strains as you would like them to be. There's also been issues arising from improper or incorrect prediction of the seasonal strains. There might be regional differences in prevalence. And of course, there are, so the need is strong, but the challenges are also great. So I've just listed a few of the challenges that the field as a whole faces. There's scientific, technical, and clinical challenges, what antigens to identify, what strategy to adopt. These are just some of the commonly used antigens that we and others have employed. There are, of course, manufacturing challenges. How do you come up, if you're developing a universal vaccine, how do you come up with the appropriate hosts and vectors? Cost is a big issue. And I guess most critically for this audience, the regulatory challenges are perhaps the largest in developing a potential universal vaccine, because there isn't a vaccine that's licensed as yet. And so, we're all working together to try to come up with frameworks and guidelines that can allow the concept of a universal vaccine to pull through. But that hasn't stopped players from entering the field and working in the field. So Invio's strategy for a flu vaccine, a broadly cross-protective flu vaccine, is based on the use of what we call synthetic consensus immunogens. And so we start out with, we've developed consensus immunogens based on the HA proteins from H1, H2, H3, H5 subtypes. And we've also developed consensus NA and NP immunogens. And so all our constructs are based on a common backbone, a common plasmid backbone, where we simply exchange out the insert of interest. To give you an idea of why this is important, so this is somewhat dated data from the WHO, where they looked at H5, cross-reactivity of H5 reference antisera against different clade 1 and clade 2 strains. And what became apparent was that while all of these diagonal elements, which are matched, clade matched anti-sera with the vaccines, with the virus strain, you get very high HI titers. The off-diagonal elements are poorly represented. So the challenge in flu, or at that time the challenge for H5, was to come up with a vaccine that allowed you to populate fairly evenly all the way across. So a single vaccine that might be able to broadly cross-protect across multiple strains. So to give you a little bit of a background on how we develop our consensus immunogens, we start out with sequences from the known strains that are deposited in a database, come up with a consensus sequence for each of the immunogens. Now this is a brute force approach because a computer can do this. But that's where the science ends and the art begins, or that's where our proprietary databases come in. So we take these consensus immunogens and then modify the sequences based on the disease information that's available to target specific pathogens. So for instance, in H5, while there are a number of known sequences that have been deposited, we chose a strategy to pick sequences to develop a consensus, only those that are jumped from birds to man, because we felt a consensus of all H5 sequences was irrelevant. So that's where some of the proprietary knowledge comes into the development and design of these antigens. We then optimize the constructs and vectors for expression and delivery, and then deliver the vaccines through electroporation. And the goal and the idea is that we get broader immunity, both T cells and B cells. Again, to give you a sense of the extent of the diversity problem in different pathogens, here's a H5 tree. And clearly, you can see that there's a reason why you have clade 1 and then clade 2.1, 2.2, 2.3 well separated, because these are really divergent strains. And if you were to choose a consensus sequence based either on a sequence alignment or a tree, it'd be hard to pick how would you pick a consensus sequence? It'd be hard to, if you chose a center of tree approach, it's not clear that the vaccine would work against any of these sequences. Well, it turns out our consensus H5 immunogen is sort of lies in the type 1, clade 1 tree, but we are able to get cross-protection against the clade 2 sequences as well. Similarly, our NA and NP antigens, again, there are different degrees of conservation and different degrees of divergence. Our NA consensus lies here on the NA tree of all N1 sequences, and likewise for the M2 and M1 antigens. I think the takeaway for this audience really is that we try to target our homologies for the consensus immunogens somewhere between 85 to 95% of identity relative to the target strains. If your vaccine antigen is more broadly divergent than about 85%, then you're likely to produce a vaccine antigen that is irrelevant to any of the strains of interest. And at the same time, if your vaccine antigen is highly, highly homologous, if you're in the 99.9% homology or identity range, then you're essentially creating a matched sequence and you lose the breadth of immune responses that you desire. So that's sort of a sweet spot that we've identified. To give you a sense of the data, here's ferret data where we showed we vaccinated these ferrets with our consensus H5 immunogen, and we can see very high HI titers against 1, 2.1, 2.2, 2.3, all the different subclades of H5N1. We see that both with HAIs, we also see that with micronewts. And again, these are very strong immune responses. In a challenge model, we did the Vietnam challenge. We were able to see protection against 100% protection against lethality with our vaccinated animals. Now, keep in mind that we also looked at individual NP-based immunogens as well. And in this experiment, we were able to show that a pure NP vaccine is also able to protect these ferrets from lethality. But nevertheless, for a variety of reasons, which we can get into during the panel discussion, we chose to move forward with an HA-containing strategy for developing a broadly cross-reactive vaccine. Here's data from our H1 consensus immunogen, where we challenged, in this case, in a mouse model, we challenged animals immunized with H1 or with the H1NP combination, challenged them with the 1918 Spanish flu. We were able to see 100% protection from lethality. Again, the NP by itself was about 50% protective in this model. Now keep in mind that the NP homology for the vaccine antigen versus the challenge strain was about 97%. The HA was only 86%. But because of the way we construct our immunogens, we are able to see protection even with a challenge that's as much as 14% divergent from our vaccine sequence. And then we've extended this concept to other, now in this case, the H3. Here's a Syncon formulation of H1 and H3 plasmids in ferrets. Again, after 2 vaccinations in naive animals, we are able to see 100% animals respond and very high titers against multiple H1 and H3 strains. If we look at guinea pigs, we then added the H5 components in here, and in this experiment, or in our experiments, we're able to see 100% seroconversion and fairly high HI titers, again, after 2 vaccinations in naive animals. So those ferrets were then challenged with the 09 strain, and then they were, again, we were able to protect the animals from lethality. So this is sort of a demonstration of the power of DNA vaccines in terms of being able to formulate multi-component vaccines to provide broadly cross-reactive immune responses and also confer protection from challenge. So we've obviously translated these data into the clinic, and so our clinical strategy for developing these broadly cross-protective vaccines, we started out with H5 primarily for historical reasons. We now have the H1 components that have been formulated. The H5 clinical trials are ongoing in the US and the Korea. We should be starting our H1 clinical trials for both the pre-09 and 09 strains to allow for broad protection against both of these strains, both of these classes of strains. That clinical trial should be starting later this year. And the goal is for us to start building additional components, add an H3 component. So H3, H5 would become a seasonal formulation that's presumably or ideally is broadly cross-protective. And then add the H2 and H7 and H9 to put together a universal cocktail. So let me switch gears to tell you a little bit about how we are applying these Syncon concepts into other infectious diseases where similar issues of divergence have been seen. So HIV, a prime example. There've been, there's a large unmet need for developing HIV vaccines. There've been 4 efficacy trials that have been conducted to date. The first 3 showed no effect. The most recent one, the RV144 study, showed about a 30% efficacy. So key considerations in HIV would be, obviously, we need improved immunogenicity, hopefully leading to improved efficacy. You need both humoral and cellular immune responses. But above and beyond all of these, there is also the issue of strain divergence. So unlike flu, where the strain changes from year to year and it rapidly spreads across the world, there is still a sense of regional distribution of HIV strains across the world. Although, in 10 years' time, my guess is this will also be a very, it might, either HIV there is a vaccine for HIV to prevent it, or the strain divergence becomes sort of broadly universal. So our strategy for HIV is to develop a synthetic consensus immunogen targeting envelopes of clade A, B, C, and D. The GAG and POL proteins are more highly conserved, and so we have a universal consensus for the GAG and POL that encompasses all of these clades. We did a comparative study with Merck and UPenn, where we compared the immunogenicity of SIV constructs using our DNA EP approach with the Adeno5 viral vector program. So if you remember, the Ad5 program went into Merck's STEP study, which was terminated in 2007 for lack of futility. But nevertheless, we looked at their Ad5 SIV vaccine in a head-to-head comparison with DNA-EP. And you can see that a viral vector, you get very strong initial immune responses, but presumably due to vector-induced immunity, you don't see continuous boosting. Whereas with the DNA program or DNA platform, you can see continuous boosting of responses. In this case, I show you the T-cell responses. You can see similar benefits if you measure proliferation, and then you can also see that both CD8 and CD4 positive T cells, both types of T cells are impacted by DNA vaccines. So it's not true to say that while conventional wisdom said that DNA vaccines were good at producing CD4 positive T cell responses, the current thinking with DNA vaccines is that when you deliver DNA vaccines with EP, you deliver a broader spectrum of immune responses where both CD4 and CD8 T cells are affected, and you also see antibody responses. So we completed a phase 1 clinical study. Here's interim data. This was in connection with in collaboration with the HVTN group under the HVTN 080 protocol, where we vaccinated subjects with a Penvax B vaccine, our Syncon Penvax B vaccine, with and without IL-12 DNA. And we are seeing very high levels of response rates amongst the vaccinees. So you're seeing induction in 70 to 85% of vaccinees developed antigen-specific T-cell responses, and these range from both CD4 and CD8. These are some of the best responses that people have reported in an HIV vaccine scenario. We're now looking at expanding on these results with additional studies. One other case where universal vaccines imply a different context is in the case of dengue vaccines. So in dengue, from a region standpoint, there's a very narrow distribution of disease, primarily within areas where the vector is co-located. And if you ask the question, what sort of a dengue vaccine would you develop in the 70s, you might think that maybe a monovalent or a bivalent vaccine might be sufficient based on what populations you are targeting. But the picture has changed dramatically over the last 25, 30 years. And now, again, like with flu, it's difficult to imagine developing a dengue vaccine that does not simultaneously target all 4 serotypes. And there are particular issues associated with induction of suboptimal immune responses against a particular serotype if you do get infected with other serotypes. So our challenge here was to develop a quote-unquote universal vaccine that really in this context means it's a tetravalent vaccine that confers protective immune responses against all 4 serotypes simultaneously. So that was a challenge for us. We are working on a candidate vaccine that takes advantage of the D3 domain of the E protein of the dengue virus. And so we developed a single plasmid formulation where we put together 4 different D3 domains consensus D3 domains from each of the 4 different dengue subtypes into a single plasmid. And here's mouse data showing induction of strong antibody titers via binding ELISA against each of the 4 subtypes. And we're also seeing pretty significant neutralizing antibody titers against each of the 4 dengue subtypes. You could argue whether titers ranging from 400 to 1,000 would be considered balanced, and that remains to be seen. But the key point is based on protection data or based on what are protective titers in people, antibody titer of around 10 to 20 is considered to be sufficient. So relative to 10 to 20, we are well above that. And current studies are ongoing to see if these levels of balance, would that be adequate for deployment in the field. And lastly, let me tell you about our HPV program, where we've again used the notion of universal vaccines in a slightly different context, primarily to access multiple serotypes of HPV. And this is a program that's now entered phase 2. So I'll share with you our phase 1 data with a cervical cancer therapeutic vaccine. Clearly, on the prophylactic side, Merck and GSK have very good options available, but neither of these vaccines is effective in women or men who've been previously exposed with HPV. Again, markets are fairly sizable. There is a clear need to develop a therapeutic vaccine. The issue with HPV is the number of different serotypes that are prevalent. 16 and 18 are clearly the dominant serotypes, but beyond that, and 16 and 18 account for over 70% of the cervical cancer cases, but they only account for 55% of the lesions. So the challenge is, what's your product going to be? Are you going to target genital warts or lesions, or are you going to target cancer? And even within cancer or lesions, depending on where you are, Africa, Asia, Europe, North America, the subdominant serotypes change in distribution. So 16 and 18 are the dominant ones, but in Africa, 33, 45 are the next ones. In Europe, 33, 31 are the next prevalent serotypes of HPV. So our strategy is that if we are developing a DNA vaccine, can we start making formulations that target either ICC or lesions, and then start combining the multiple serotypes into a single formulation? And others are also doing this. I just heard, Merck's making a 9-valent HPV vaccine for prevention. So the concept is similar. The one difference would be that with DNA, formulation is as easy as combining the plasmids into a single vial. Of course, the technical challenge with DNA is, can you get DNA manufactured at a high enough concentration that you can combine 3, 4, 5, 6 different plasmids into a single dose. And this is something that I'm happy to say that our colleagues at VGXI have been able to solve, where we've been able to make formulations in excess of 10 mg per ml. And that allows us to put in as many as 10 different plasmids if 1 milligram per milliliter is an intended dose for your vaccine. For the HPV product, we've gone as high as 6. We have other formulations, particularly in HIV, where we've accessed the full range of our 10 to 15 milligrams that we're able to achieve. Just some top-line data on the HPV phase 1 study. What we observed, this was a 3-dose dose escalation study. There were 6 subjects per cohort. These were women who had a previous history of SIN 2-3, and then they were otherwise currently, but they've been treated for their SIN 2-3 lesions. So we're able to show that we see a dose-dependent increase in T-cell responses. Our vaccine formulation contains 4 antigens, E6 and E7 proteins from both HPV subtype 16 and HPV subtype 18. In this formulation, we're able to show induction of T-cell responses against all 4 antigens. Response rates, we saw T-cell responses in 13 of 18 vaccinated subjects. So these women have been previously exposed to HPV. So to the extent that there may or may not be issues on immune tolerance, we are able to overcome them with our vaccine. Significantly, we are also able to show a very strong antibody response against the immunogens in upwards of 80% of the vaccinated cases. So let me just finish by, again, sort of bring this back together in terms of a product development story. Inovio is focused on developing vaccines. We are a vaccine product development company. We have a number of different programs ongoing in cancer, as well as infectious diseases. And then our R&D pipeline, we're looking at not only endemic or chronic infectious diseases, we're also looking at emerging pathogens to try to target some of the greater targets of unmet need. We have several development partners. We are grateful to MVI and NIH for funding the malaria program, NIH for funding the HIV vaccine development program, and we have and our industrial partners for helping carry the ball in terms of adopting the technology in their product development as well. So in conclusion, what I'd like to say is that, vaccine product development, like any product development, is really, requires a concerted development of formulations, the content, as well as delivery. It's not just a case where you can take any DNA piece and then inject that and expect that to be immunogenic. One does need to pay attention to what are your target immune responses. Are you looking for predominantly neutralizing antibody humoral-based responses or cellular immune responses? And if, depending on the answer, what are the correlates? What are you trying to achieve? And depending on the answer, product development is focused around the answers that you seek to address. We've shown that immune phenotypes are diverse and manipulatable. We can target multiple pathogens and disease states. We have shown we can impact disease in challenge models, disease or infection in a number of different challenge models. And our clinical data from prophylactic HIV and therapeutic HPV looks promising. And we are, I guess, stay tuned for the flu data that we should be getting, in the near future. And lastly, I put this up as, not only to acknowledge all of the people within the company and outside of the company, but really to underscore the point that vaccine product development is an expensive proposition. And in particular for a biotech company without the deep pockets, we have to build the bridges across academia, CROs, collaborators and affiliates, as well as the health agencies. The neutralization and HAI data that I showed you for the H5, that was done in collaboration with Hannah Golding's lab at the FDA. They've built the expertise to develop assays and report results. So we are happy to collaborate with them to help move these programs forward. Finally, I do want to acknowledge our clinical volunteers and sites, as well as notably funding from a large number of government and non-government organizations. And thank you.